Alexander Fleming predicted the rise of antibiotic resistance before it had even started. It's now no secret that we need more antibiotics. No new antibiotic drug classes have been discovered since 1980. In recent years, researchers have been turning to nature to try and find novel antimicrobials. Extreme environments like the deep sea, or plants and animals like lichens and ants, have all yielded hopeful results. But if there are all these potential new antibiotics, why aren't they on the market? Every potential new drug must go through a plethora of tests, trials, and approval boards before it can be administered to a patient in a clinical setting, and most of them fail. So how are new medicines developed? Once the researchers have a lead compound, like a metabolic product from a fungus or bacteria, they have to find out how it might affect the patient. Is it toxic? Can it be metabolized in humans? And how much of it is needed for it to be effective? Depending on the answer to any one of these questions, it could be decided that the compound won't make a suitable drug and development should be stopped. Then it's back to square one. However, if the development of a compound is taken forwards, then it goes into the chemistry, manufacturing, and control stage. Here, the physical characteristics of the compound are determined, like its chemical makeup and whether it's soluble and stable. The scientists can then decide how this new drug will be manufactured and administered, for example, orally, injected into the muscle, or intravenously. During this stage, it's also important to evaluate costs. If it's too expensive, or researchers can't find a way to safely administer the compound, then development will cease. If the compound still seems to be suitable, then safety tests begin. Different organs may respond to a drug in different ways, and so it's essential to test the compound in as many ways as possible to determine whether it can be given to people safely. This stage requires stringent testing on cell culture, cells grown in a lab that can show how cells in people would react to the drug, and later in animals to examine how complex biological systems might respond. If the information gathered during these preclinical tests suggests that the compound would be a safe, effective, and economically viable drug, then the development process can go on to the next step. Clinical trials for new drugs are often split into 4 phases and can take many years to complete. As the drug progresses through the stages, the number of participants in the trial increases. The first 3 phases determine dose safety levels, effectiveness, commercial value, and potential side effects. If the drug passes, it goes through regulation review and, if approved, can be produced and marketed. Even when the drug has obtained approval, the drug testing hasn't finished. Phase 4 watches the drug's use in public for a minimum of 2 years. This phase is designed to detect any rare or long-term adverse effects that wouldn't be possible to detect during the previous phases. The entire process from preclinical research to marketing can take up to 18 years and is incredibly expensive. It's not uncommon for the process to cost well over $1 billion. However, despite the time and money, antimicrobial resistance is a growing threat, and it is vital that scientists continue the search for the next breakthrough.